Welcome back to this next video, and in this video we are going to talk about the fibroblast growth factors, about the fibroblast growth factor receptors, what are the fibroblast growth factors and what are the fibroblast growth factor receptors, and we'll be focusing on one of the important pathway that is known as the MAPK signaling pathway that is actually mediated by the fibroblast growth factors and its receptor. So the first thing is, what are fibroblast growth factors? The fibroblast growth factors, that is a family of the cell signaling proteins. That means that they are involved in the process of the cell signaling, and they are actually making a connection between the cell and its extracellular environment. And the members of the fibroblast growth factors, they are involved in the process of angiogenesis, which is actually the physiological process through which the new blood vessel they form from the pre-existing vessels. Its members are also involved in the wound healing, in the embryonic development. The members also act as the mitogen, and they are involved in various endocrine signaling pathway. So the pathway that we'll be focusing in this video, that will be actually as an example of the fibroblast growth factor acting as a mitogen. If you talk about the fibroblast growth factors, they are the heparin binding protein. So what I mean by this heparin binding protein, if this is the gene for the fibroblast growth factor, the gene is going to get transcribed and translated into the protein. The protein is going through the process of post-translational modification, and in this process of the post-translational modification, the heparin moiety, which is actually a carbohydrate moiety, that is going to get attached to this particular protein. So this complete this protein plus this heparin part, this is actually known as the fibroblast growth factor. So these fibroblast growth factor, they are the heparin binding protein. One important thing that is known as the heparin sulfate proteoglycan, that is a complete different set of proteins. The proteoglycans, they are actually proteins, they are heavily glycosylated, and these are very important for the interaction of the fibroblast growth factor to its receptor, which is known as the fibroblast growth factor receptor. Now, how you will differentiate between the heparin and the heparin then? Because this is a different protein, this is the fibroblast growth factor, this is a totally different protein which is known as the proteoglycan, but they contain this part. One is known as the heparin and the other one, they are known as the heparin. Now this heparin and this heparin sulfate, they both belong to the class known as the glycosaminoglycans, which is actually a family of the carbohydrates. Now the major difference between the heparin and the heparin is that the common disaccharide units between the heparin and the heparin, they are different from each other. If you talk about the heparin sulfate, which is actually present in these proteoglycans, and if you talk about its common disaccharide, that is made up of the glucuronic acid, and that is made up of the N-acetyl glucosamine. So these two, they are going to make a disaccharide, which is the building block or the basic unit of the heparin sulfate. If you talk about this heparin, which also belong to the family of the glycosaminoglycan, so in the heparin, the common disaccharide unit, that is made up of the 2-O-sulfo alpha L iduronic acid, which is actually represented by this particular part, and that is made up of the 2-deoxy 2-sulfoamido alpha D glycopyrenosyl. They are the common disaccharide unit in the heparin. So the heparin and the heparin, both of them, they belong to the class of the glycosaminoglycans, but their disaccharide unit composition, that is different from each other, and that is actually used to differentiate between the heparin and the heparin. So the fibroblast growth factor, that is going to interact with its particular receptor known as the fibroblast growth factor receptor, but this interaction needs the help of the heparin sulfate proteoglycan. So this will be clear in the next slide, how the heparin sulfate proteoglycans, they are helping in the interaction of the fibroblast growth factor to its particular receptor. Now this fibroblast growth factor, which I've told you is a large family of the polypeptide growth factors, they are found in organisms ranging from the nematodes to humans. So you can theoretically find these fibroblast growth factors in a wide variety of the living organisms. If you specifically talk about the vertebrates, 22 members of the fibroblast growth factor family, their molecular weight range from 17 to 34 kilodaltons, and they actually share between 13 to 71% amino acid identity. If you specifically talk about the human beings, so far 8 fibroblast growth factors have been identified in humans, and they are named as fibroblast growth factor 1 to 7. By that I mean that the fibroblast growth factor 1, fibroblast growth factor 2, 3, 4, 5, 6 and 7, and then the fibroblast growth factor 9. Now this fibroblast growth factor 8, they have not yet been identified in the human beings, and that is only identified in the mice. Now member of the fibroblast growth factor 1 through fibroblast growth factor 10, they all bind to the fibroblast growth factor receptors. This fibroblast growth factor 10, you can see that this is not present over here, so we are talking about the vertebrates over here. Now that means that all of the fibroblast growth factors, they bind to the fibroblast growth factor receptors. Now most of these fibroblast growth factors, they are translated with signal sequence. We'll be talking about the importance of the signal sequence in a while. And the fibroblast growth factors, they have got this signal sequence, and they undergo post-translational glycosylation, as I've told you, in the form of the heparin, which is characteristic of the typical secreted proteins. If you talk about the structure of the fibroblast growth factor receptor, this is actually a general structure of the fibroblast growth factor receptor. The first important part of this particular fibroblast growth factor receptor, keep in mind one thing, if we are talking about single protein starting from the N terminal up to the C terminal. So we are not talking about a complex of the proteins, it is a single protein with a different kind of the domains present in this particular protein. So the first important thing that you can see over here is the signal peptide. Now the function of the signal peptide is to, for a cell, is to translocate the protein usually to the cellular membrane. As you are talking about the fibroblast growth factor receptors, and you all know that we need these receptors on the surface of the protein. So these signal peptides, they are actually going to help move the fibroblast growth factor receptor from the cytoplasm, where it has been translated, to the cell membrane, where it is required to function. So the first important part, that is the signal peptide. Another important region in the fibroblast growth factor receptor, that is known as an extracellular ligand domain. And the extracellular ligand domain, it starts from here. If you can see this D1, this acid box, this D2 and D3, this whole part or this whole domain is actually making the extracellular ligand domain. This extracellular ligand domain, if you can see in this particular diagram, is made up of 3 immunoglobulin-like domains. One is known as the D1, and the other one is known as the D2, and the last one, that is known as the D3. They are called as the immunoglobulin-like domain because they share their structural similarity with the immunoglobulins. So in the extracellular ligand domain, you are actually going to find these 3 subdomains, the D1, the D2 and the D3. Now the function of this extracellular ligand domain, as you can see from the name, that this is the ligand domain. So this is the region where the fibroblast growth factor, it binds. So this is the actually the binding region for the fibroblast growth factor. Another important domain, that is known as the transmembrane domain, and in the fibroblast growth factor receptor, you usually find a single transmembrane helix domain, and it is present over here. Now the intracellular domain, it is actually present with a tyrosine kinase activity. So if you can see over here, this part up to here, the extracellular domain that will be present on the surface of the cell. That means that will be present outside the cell in the extracellular environment. This transmembrane domain is present within the cell membrane, and this intracellular domain that is actually present in the cytoplasm of the cell. So this intracellular domain, it have got this tyrosine kinase activity, and there are two different domains. One is known as the tyrosine kinase 1, the other one is known as the tyrosine kinase 2, and both of them, they have the function of the tyrosine kinase. Keep in mind one thing, that this tyrosine kinase, they are known as the tyrosine kinase because when these kinases, they interact with their substrate protein, they are going to phosphorylate the tyrosine residues in its target protein. The kinase, they are the protein which have the ability to phosphorylate its particular substrate, but when you call it as a tyrosine kinase, this means that this particular kinase is going to phosphorylate the tyrosine amino acid in its target protein and not any other amino acid. And this is the last part, the C terminal of this particular protein. So this is the general structure of the fibroblast growth factor receptor with important region, the extracellular domain, the transmembrane domain, and the intracellular domain with signal peptide. Now, if you talk about the extracellular domain over here, if you can see over here, the three immunoglobulin like domain, the D1, the D2, and the D3, they are present. These D2 and D3, they are actually the region where the fibroblast growth factors, they are going to bind over here. In this particular diagram, if you can see the D2 and the D3, they are actually the regions of the receptor where the fibroblast growth factors that can actually interact utilizing its heparin domain. This is the transmembrane helix, and this is the structure of the kinase domain that we just discussed, highlighting the important regions in this particular kinase domain. Two of the regions, they are very important. One is this activation loop. Once you have this activation loop is actually responsible for the activation of this kinase domain, and this catalytic loop is responsible for the phosphorylation of its substrate proteins. Now this diagram is actually showing you the importance of the heparin sulfate proteoglycans. If this is the fibroblast growth factor receptor, this one is the heparin sulfate proteoglycan. So the fibroblast growth factor, as you can see this with this particular ball, that is actually sandwiched between the fibroblast growth factor and the heparin sulfate proteoglycan, and this heparin sulfate proteoglycan is actually facilitating the interaction of the fibroblast growth factor to its particular receptor. Now imagine if this heparin sulfate proteoglycan is not present, the interaction between the fibroblast growth factor receptor and the fibroblast growth factor, that is going to be highly compromised. So you need the heparin sulfate proteoglycan for the proper interaction of the fibroblast growth factor receptor to its particular ligand, which is the fibroblast growth factor. If you talk about the fibroblast growth factor genes, the fibroblast growth factor receptor genes, there are 4 genes, but the natural alternative splicing, it is going to produce 46 different isoforms of the fibroblast growth factor receptor. So you have got 4 genes, you have got 48 different isoforms of the fibroblast growth factor receptor, and this is because of the natural alternative splicing. Now these isoforms, these 48 different isoforms, they vary in their ligand binding properties and their kinase domain. The ligand binding properties, they are very important, because if they are the same, that means only a single kind of the fibroblast growth factors that will be interacting with these receptors. But as these ligand binding properties, they are different from for the different fibroblast growth factor receptors, that means they can interact with a variety of the fibroblast growth factors. As we discussed, that there are about 22 different fibroblast growth factors so far being identified in the vertebrates. However, they all share the common extracellular region, which is composed of the 3 immunoglobulin like domains, and thus they all belong to the family of the immunoglobulin superfamily. So in all of these fibroblast growth factor receptor, you are going to find these D1, D2 and D3. However, their properties to binding to the ligands, that is variable. One important thing is that how these fibroblast growth factor receptors they are regulated. Now, one of the important regions that is present between the D1 and the D2, that is known as the acid box, that has been represented over here. This is the D1 domain of the extracellular domain. This is the D2 domain of the extracellular domain, and between the D1 and the D2 there is this acid box. Now, this is called as acidic box because if you look at the amino acid composition over here, they are actually made up of a lot of aspartate and the glutamate, which are the acidic amino acid. Therefore, this region is known as the acidic box. Now, this acidic box, it is very important for the regulation of the activity of the fibroblast growth factor receptors. And how this the regulation mean that when you want to turn on the receptor and when you want to turn off the receptor. So when there is no fibroblast growth factor, that means that the fibroblast growth factor receptor that should be turned off. And when there is fibroblast growth factor, that means you should turn on the fibroblast growth factor receptor, and this function is carried out by this acidic box. Now, what this acidic box do, that it is going to interact with the heparin sulfate binding site to prevent the receptor activation in the absence of the fibroblast growth factor. As you can see over here, the D2 and the D3, they are actually the fibroblast growth factor binding region, and the part of the fibroblast growth factor that actually interacts with this D2 and D3, that is the heparin sulfate part. So what this acidic box do, that it is going to loop over here on the D2 and the D3, and when the acidic box it loops on the D2 and the D3, that means nothing can bind to the receptor, that nothing can bind to this particular receptor, and hence you cannot activate the receptor. So this acidic box, in the absence of the fibroblast growth factor, is going to cover the heparin sulfate binding site of the extracellular domain, thereby preventing the attachment of any other thing to this particular receptors. Now, this immunoglobulin like domain D2 and the D3, as I've told you, that is sufficient for the binding of the fibroblast growth factors. Now, each receptor that can be activated by several fibroblast growth factors, and in many cases the fibroblast growth factors, they can activate more than one receptors. For example, if you talk about the fibroblast growth factor 1, it can bind to 7 principal fibroblast growth factor receptor. There are certain cases where a single fibroblast growth factor can only interact with a single kind of the receptor. For example, the fibroblast growth factor 7, that can only interact with the fibroblast growth factor receptor 2b. Now, how this fibroblast growth factor and the fibroblast growth factor receptors, they are going to activate a particular pathway. So this is just an overview of the pathway that we'll be discussing in the detail in the next slide. So the first thing is that the fibroblast growth factor, it binds to the receptor, and when it binds to the receptor, it is responsible for the dimerization of the receptor. As you can see over here, there are 2 receptors. This is, say for example, the receptor 1, and this is receptor 2. So when there is no growth factor, there is no fibroblast growth factors, these 2 receptors, they are away from each other. But when the growth factor, they binds to these receptor, it is actually responsible for the dimerization of the receptor. Now, when the receptor, they dimerize, as you can see over here, their intracellular domain, they are going to come in interaction with each other. This one is the extracellular domain, which is present outside the cell membrane. The domain present inside the cell membrane is the transmembrane domain, and the one present inside the cell is known as the intracellular domain. So when these receptors, they come close to each other, the intracellular domain, they are actually coming in interaction with each other. As this intracellular domain is tyrosine kinase domain, that means it has the ability to phosphorylate the tyrosine amino acid in its substrate protein. This one is a tyrosine kinase domain. It has the ability to phosphorylate the tyrosine amino acid in its substrate protein. So what these receptors do, they are going to phosphorylate each other. This tyrosine kinase domain is going to phosphorylate this tyrosine kinase domain on the tyrosine amino acid, and this tyrosine kinase domain is going to phosphorylate this tyrosine kinase domain on the tyrosine amino acid. So if you can see over here, the binding of the fibroblast growth factor is actually making the interaction between the intracellular domain or the tyrosine kinase domain of these receptors, and thereby activating them sorry, by the addition of the phosphate groups on the intracellular domain. Now when the phosphate groups, they are attached to the intracellular domain or the tyrosine kinase domain, different kind of the proteins, they are going to interact with this phosphorylated tyrosines now. One of the important protein that interacts with this phosphorylated tyrosine is known as the GRB2. Now how this GRB2 that interacts with this phosphorylated tyrosine? Now this GRB2 contain an important domain which is known as the SH2 domain. As you can see over here, that the GRB2 contain a particular domain which is known as the SH2 domain, and this SH2 domain, it has the ability to bind to phosphorylated tyrosines. Not simple tyrosine, but when the tyrosines, they are phosphorylated, this SH2 domain has the ability to bind to the phosphorylated tyrosines. And as here it is going to find a phosphorylated tyrosine, the GRB2 is going to bind over here. Now when the GRB2 utilizes its SH2 domain to bind to the phosphorylated tyrosine, and the GRB2 interacts with this phosphorylated tyrosine, structural changes are taking place in the GRB2, and when these structural changes are there, the GRB2 now interacts with another protein which is known as the SOS protein, or which is known as the son of 7-less protein. Now this SOS protein is actually a GEF protein, and we will discuss that in a while what this GEF protein means. At this particular time, you should keep in mind that this SOS, which is actually serving as GEF, has the ability to replace the GDP with a GTP. So when the GRB2 interacts with the phosphorylated tyrosine, it is going to interact with the SOS protein. The SOS protein is going to get activated, and this SOS is going to interact with the RAS protein. Now if you look at over here, initially the RAS protein is in interaction with the GDP. So when the SOS it interacts with the RAS with a GDP, it is going to convert it into the RAS GTP. So the SOS is going to replace this GDP with a GTP. So when the RAS GDP it is converted into the RAS GTP, the RAS GTP is now in the active form, or you can say this is the active form of this RAS protein. One thing to keep in mind that is this RAS is a membrane bound protein. As you can see over here, they have got this lipid moiety, and with the help of this lipid moiety, it is in interaction with the cell membrane. Now when the RAS it is converted into the RAS GTP form, which is the active form of the RAS GTP, the RAS GTP is then going to phosphorylate another protein which is known as the RAF protein. As you can see over here, after the activation of the RAF has been converted into the phosphorylated form. This RAF is the rapidly accelerated fibrosarcoma. This is the complete name of this particular RAF protein, and in some of the literature you will find that this RAF is also known as the MAPKKK. So both of them, they are referring to one and the same thing, the RAF or the MAPKKK, and that is one and the same thing. That is the protein which is activated by the RAS protein. Now when the RAF protein it is phosphorylated, it gets activated, and this RAF is going to activate another protein which is known as the MEK1 or the MEK2. This MEK is the mitogen activated kinase 1 or 2, or it is also known as the MAPKK. When the MEK it is activated, it is going to activate another set of protein which is known as the ERK12, and this ERK is the extracellular signal regulated kinase, or it is also known as the MAPK. And this is because of this ERK or the MAPK that this pathway is known as the MAP kinase pathway. Now, if you look at this one, we are actually activating a series of the kinases. The RICE GTP is activating a RIFE protein, and this RIFE protein is a kinase. So when it is activated, it's going to phosphorylate its substrate protein. The substrate protein is MEK102. Again, this MEK is a kinase. It is going to activate the ERK, and this ERK again is a kinase, and this ERK is going to move into the nucleus of the cell. And when it moves into the nucleus of the cell, it is going to activate those particular proteins or those particular genes which are responsible for the proliferation, the growth, or the survival of the cell. For example, if you talk about the growth and the proliferation, this ERK2 protein are going to activate the cyclin and the CDKase genes. And when the cyclin and the CDK genes they are activated, that is going to activate the cell division pathway, or that is going to start the cell cycle. Now I want to talk about this RICE protein, because that is one of the important protein in all of this particular pathway. Because if you see over here, the activation of the RIFE, that is actually the one of the important step in the activation of this kinase cascade. So this RICE protein, they are actually a very large family, this RICE superfamily, and they contain a family of the monomeric GTPases. Now you need to understand this GTPase. Now these GTPases, they bind to guanosine triphosphate and they hydrolyze it to the GDP. That means that when the GTP is bound to them, they have the ability to convert them into the GTP. Now this GTP binding and hydrolysis in these particular RICE protein, this activity is taking place in a highly conserved domain which is known as the G domain. And this G domain is actually made up of 166 amino acid, and its molecular weight is 20 kilodaltons. If you talk about the cell signaling from this RICE family, only the RICE and the Rho families, they are actually responsible for relying the signal from the cell surface receptors by interacting with a different intracellular signaling protein. And as we have seen over here that the RICE is interacting with the MEK directly, it is indirectly also interacting with the ERK proteins. If you talk about the human beings, there are three major closely related RICE proteins which are known as the H, K, and the N-RICE. Although these different kind of RICE, they have several different functions, they are thought to work in the same way, and they are collectively known as the RICE proteins. Now, how this RICE protein is important in this particular pathway? What we do usually in genetics is the mutation studies or the inhibition studies. So if you inhibit the function of the RICE by the micro injection of neutralizing anti-RICE antibodies, you can simply say if you are injecting those particular antibodies which inhibit the function of the RICE, the cell proliferation or differentiation response normally induced by these activated receptor tyrosine kinase that do not occur. Now this receptor tyrosine kinase, it is just a fancy name for the fibroblast growth factor receptor because the kinase domain in these particular receptor are the tyrosine kinase, so therefore they are known as the receptor tyrosine kinase as well. So the receptor tyrosine kinase, the fibroblast growth factor receptor, you are referring to one in the same thing. Now conversely, if these RICE proteins in 30% of the human tumors, these RICE proteins, they are hyperactive, and when they are hyperactive, they are actually going to contribute to the uncontrolled proliferation of the cancer cells. Now this RICE protein, as I've told you, that actually work as a molecular switch, or you can say the RICE protein, it occurs in two different conformational states. This RICE protein is active when it is bound to the GTP, and it is inactive when it is bound to the GDP. Now, how the rice protein is achieving these two different conformational states. So, two different classes of the proteins, they are responsible for switching of the rice protein into the rice GDP and into the rice GDP form. One of the important protein is known as the rice guanosine nucleotide exchange factor, the rice GEF. As I've told you in the last video, in the last slides, that the rice GEF and the SOS, they are actually one and the same thing. So the SOS is an example of the rice GEF. Now what this rice GEF do, as you can see over here, that initially the rice is in interaction with the GDP, which is the inactive form of the rice. So when the GEF, it interacts with the rice GDP, it is going to replace this GDP with a GTP, and after the interaction with the GEF, now you have the rice in the GTP bound form. Now, once the activity of the rice protein is over, you want to convert the rice GTP into the inactive form, that is the rice GDP, and this particular function is performed by another protein which is known as the rice GAP, or which are known as the rice GTPase activating proteins. Now, the hyperactive mutant forms, which I've told you over here, that in about 30% of the human tumors, they are hyperactive. Why they are hyperactive? Because these hyperactive mutant form of the rice, they are resistant to the GAP mediated GTPase stimulation. Now that means once the GEF has converted the rice into the GTP form, as these rice are no more interacting with the GAP, they will not be converted into the inactive form, and when they are constitutively in the active form, that means that is going to go for the development of the cancer. Now this is a very complex diagram. I am not going into the detail of this, but I just want to show you that the different positions of the tyrosine that can be phosphorylated by the tyrosine kinase domain. As you can see over here, the 583, the 585, the 653, the 654, a lot of the tyrosine, they are phosphorylated, which are actually helping in the interaction of the GRB2. So I hope you have got a good idea of the fibroblast growth factor and its receptor and how this particular pathway works. If you like the video, please hit the subscribe button, like this video, and hit the notification bell. We will continue this in the next video.